third and final part of the Chapter 9 DNA structure and replication chapter deals with the actual process of DNA replication and polymerase chain reaction, otherwise known as PCR. And so the learning outcomes for this section are that we're going to demonstrate the semi-conservative mechanism of DNA replication, look at the steps of DNA replication and the enzymes that are involved, and then really explain the process of polymerase chain reaction and how it can quickly make copies of DNA. So DNA replication is considered semi-conservative. What that means is two identical double helixes are formed from one original parent double helix. In other words, each new DNA double helix conserves half of the original. Okay, that's where the semi-conservative, because it's conserving part of it. And that individuals that kind of did research on this to show that DNA replication is semi-conservative were Meselson and Stahl. And they demonstrated the semi-conservative mechanism of DNA replication by using different isotopes of nitrogen, a heavy form and a lighter form, to kind of trace the pattern to show where it was distributed. So they used this higher density nitrogen, incorporated it into one strand of each of the daughter double helixes. And so if this is the parent molecule and these are the daughter molecules, basically what they were able to do is show by labeling one of the strands with the parent molecule with the heavy molecule, then they could see that the newly copied strand was this lighter one, and it still conserved part of the original one that had the heavy nitrogen in it. So it was kind of a way for them to be able to trace and keep track of what was actually passed on to each of the daughter cells. So the steps of replication, first of all, just as a review, remember that DNA replication occurs during the S phase of interphase, during the cell cycle, prior to cell division. So every time your cells are getting ready to divide, they need to replicate their DNA first so that each new cell will receive a copy of that DNA. And so the first step and enzyme involved is an enzyme called helicase. What it does is it unwinds and separates the DNA by breaking the hydrogen bonds that are holding those base pairs together. And the site where the DNA gets opened is called the replication fork. So we often compare DNA to like a zipper. Like if you unzip a zipper, it separates into the two halves, right? Well, it's more like a broken zipper. You know how sometimes your zipper kind of opens up in the middle spot of your middle of your jacket? Well, you have lots of different replication forks of different areas where the DNA will open up and start replicating along it. Otherwise, it would take too long to go all the way from one end of the DNA to another. It gets opened up at multiple spots to be copied. But because DNA wants to keep winding back up again, it has the tendency to stay in that helix shape. Some binding proteins do help hold the two strands apart. Okay, and then from there, another enzyme called primase adds a short RNA primer to the DNA template strand. And this is needed for the DNA polymerase to start. DNA polymerase can only start putting down new nucleotides if there's already a portion of the nucleic acid there to kind of get it started. And so that little piece of primer made from the RNA helps get that DNA polymerase a place to start and build off of. The big enzyme that does most of the work here is DNA polymerase. Its job is to add those DNA nucleotides to that already started RNA primer, and then it moves down the open DNA in a 5' to 3' direction, adding nucleotides according to the correct base pairings. A with T, C with G. Okay. Now DNA polymerase will also proofread, and then along with the exonucleases, and check for incorrect pairings as well as remove that RNA primer, because we can't just keep a chunk of RNA stuck inside the DNA. So eventually, as the DNA polymerase is proofreading and the exonucleases kind of help with that proofreading, the exonuclease can remove the RNA primer so that the DNA polymerase can put down the correct DNA bases. And then finally we have ligase, and that seals up the sugar phosphate backbone, makes sure all those bonds are bonded together to make the sugar phosphate backbone. So let's take a look at what that looks like. First of all, you have your parent molecule, it's opening up, and you can see that the new nucleotides of the daughter molecules are being added to the sides there. Each open strand provides a template for the daughter strands. This is a review of those enzymes that we were talking about. The helicase that unwinds and breaks those hydrogen bonds, unwinds the DNA, the binding proteins that stabilize and hold them apart, the primase that adds the short primer, RNA primer, the DNA polymerase that binds those new nucleotides, and then the ligase that joins the Okazaki fragments together. And then sometimes we talk about an exonuclease. It works along with the DNA polymerase to remove the RNA primer. We got to get this chunk out of there eventually. Okay. So here you can kind of see it in a visual format. Here, this is the helicase right here. And that's breaking those hydrogen bonds and separating the strands. And then you have the binding proteins holding the strands apart. And then you have DNA polymerase. And then, of course, it's oops, no, I got that wrong. Sorry. I'm jumping ahead of myself. You have the primase that's putting down this short little piece of RNA primer there. Now you have the DNA polymerase. And again, notice it's only going in a 5' to 3' direction. So it's going, you have a kind of like two cars going opposite ways on the road. You have that one polymerase going that direction, another polymerase going this other direction. And it's going to not only put down the new nucleotides, but it's also going to proofread and replace incorrect bases. Eventually that, along with the exonuclease, will remove that little primer too. So you have a continuous strand that's going, and then you also have some broken up fragments from wherever those primers are located that kind of interrupt things. Those are called Okazaki fragments, where they're short little pieces that have to get sealed up later on. And then it talks about the enzymes removing the RNA primers. So those could be either exonuclease or DNA polymerase. Sometimes takes care of that as well, as it's proofreading. And then ligase, that's what this one is referring to, comes along and seals up any of these open gaps, any of the sugar phosphate backbone. Again, if we only started at one end of your DNA and had to go all the way straight through to the other, that would take forever. And so instead, the chromosome is replicated at hundreds of points simultaneously. And we call those replication bubbles. And then the pieces just kind of get joined together that have been replicated. And you've got your double helix made. Two copies right here. Now, we have made great strides in the study of DNA and technologies with DNA, and one of them is what's called PCR, or polymerase chain reaction. It was discovered by Carey Mullis in 1983. Yeah, that's when I was probably about, sophomore in high school. And then 10 years later, he won the Nobel Prize for it. But it's a method to exponentially amplify DNA. In other words, make millions of copies of DNA in a very short amount of time. So a very cool process. You start out with your single copy of DNA template, and it goes through a whole bunch of cycles, and you can see how many billions of copies you can make in a short amount of time. These are the main ingredients you need, and I'm going to focus really on these first four because the water and buffer just kind of help in terms of making the solution that allow you to go through the process of PCR. But you need your template DNA. That's your target DNA that you want to have copied. Then you need to have primers. These are those short little pieces that allow the polymerase to work. Okay? And then you have a special kind of polymerase called Taq polymerase. It's called that because it came from a bacterium called Thermus aquaticus that normally lives near hot springs. And it's stable at high temperatures. If we would start heating up your DNA, it would denature and it'd be destroyed and it wouldn't be able to get copied. But because we use a polymerase that comes from that heat-resistant bacterium, now we can do the temperature conditions of PCR and the enzyme will remain stable. So that's the one that's going to act as our DNA polymerase. And then finally you need all of your bases, right? Your A, T, C, and G. Okay, and those are called the D and NTPs, nucleotide bases there. So those are the components you put into a little container, and you mix that up along with your water and your buffer, and then you load it into a thermocycler, and I'll show you a picture of a thermocycler in a minute, but there's three main steps that get repeated over and over and over for about 30 to 32 cycles. The first one is called denaturation. This is when the DNA template gets denatured with high heat to separate the strands. Okay, we're not trying to destroy it completely. We're just heating it up enough to separate it into its two strands. Then we have annealing, and that's where the primer is going to get added and bind to the complementary sequence on the DNA. And then extension is when the DNA polymerase is going to go down, putting down the correct base pairing, and create a new strand of DNA complementary to the template DNA. All right? And so this is what a thermocycler looks like. So you take your little container that has all those things we mentioned earlier that need to go in there, and you're going to denature at 94 degrees. See how hot this temperature is? And then it gets cooled. And then the temperature goes up again, and so on. And it'll just keep going through that cycle over and over and over again for 32 cycles usually. How could we use PCR? Why would we want to amplify it? Sometimes there's not enough DNA available left at, say, the scene of a crime, and we needed to make much more copies so we could actually do the tests on it. So we can look at inherited diseases. We can use it to identify disaster victims if there's not much left behind and we only have a little bit to go on. We can use that to amplify their DNA, figure out who the mom or dad is of a child, paternity testing, identify a crime victim or perpetrator. You can look at digestive tracts of carnivores to reveal what kind of food they ate. Transmissions of disease, like Lyme disease or something like that. So all sorts of different uses for that. So it's just kind of a nice technology. We'll do this more in the spring when we get to our biotech units, and you'll get to do some PCR. You're going to amplify your own DNA. We're going to get that from your cheek cells.